okay? There's a lot of things that C3A so there's a lot of things that C3A can do. We're not going to go through all the list of them, right? But the A parts, the C4A is going to do this. The C5A is going to do this. Does that make sense? So the A parts are stimulating inflammation and phagocytosis. The B parts are just keeping this cascade alive. Okay? Now, at some point, we get to the MAC complex, okay? So what happens is if you activate enough proteins, so see how it's 5, 6, 7, 8, and 9 right here? These the B parts, once they activate and stimulate the next protein, the remaining components are going to stick together. And what they do is they insert themselves into the membrane like this. Do you guys see it forming this hole in the membrane? Remember we saw this before in natural killer cells, and we saw this in the lymphocytes. It's the exact same forming a hole. This hole is called the membrane attack complex, which is abbreviated MAC. Okay? So, if we look at this right here, we're saying that each pathway converges on C3, which breaks into C3A and C3B. The B is going to stimulate all of those proteins until you get that membrane attack complex called the MAC. The C3A is what's going to go through and stimulate inflammation. Okay? Any questions on this? So, like when it says right here that this is started by antibodies, that's done through IgM. Okay? Any questions on complement before we move on? Okay. Now, the only thing significant about IgA is this is usually found in secretions in the body. So you're going to find it in your sweat, you're going to find it in your tears, you're going to find it in your saliva. So this is basically coming from like your glands in the body, and it protects against surface pathogens. So obviously your skin is on the surface, so if you secrete IgA on the surface, you're going to fight on the skin. If it's in your tears, it's going to fight in your eyes, right? If it's in your saliva, it's going to fight in your mouth. If it's in your mucus, like your respiratory tract, it's going to fight in your trachea. But the whole purpose of IgA is surface protection. Okay? Now, IgD, they don't know very much about it other than the fact that it is your B-cell receptor. Okay? So, let's go back to this picture right here. We said that this is an antibody, and it's your B-cell receptor. It's specifically an IgD. Does that make sense? Always?
Always.
Yeah.
That's its job.
Yeah, that's it. And that's the only thing they know about
Whenever
it.
it's a B cell, it's IgG.
Okay, now I'll come back to IgG in a minute. IgE is what binds to basophils and mast cells. What did we say a basophil or mast cell's job is? They do the same thing. They release histamine, okay? Now, what happens is that if you have some sort of, so this is in your tissue, like you have, I'm just gonna do a little drawing right here, okay? If you have a little bacteria that comes in here, okay? Then the bacteria gets surrounded, or like your antibodies bind to the bacteria. This is your IgA, okay? The mast cell sees and recognizes this IgA right here, and when it sees and binds to that IgA, then that's when it releases your histamine. Yes, sorry. Okay? So, when the IgE's whole purpose is to activate the mast cells and your basophils in the first place. And remember we said that your histamine is what causes the whole inflammation process to happen, right? So, like at the very, very beginning, when we said inflammation, right? We said that you have your like blood vessel. The first thing is we make this blood vessel wider, and then we increase the permeability. We open gaps in your blood vessel wall, right? What caused this to happen in the first place was you said that your mast cells had to secrete histamine, right? But your mast cells aren't going to just secrete histamine randomly. It specifically has to find this IgE. Okay? Now, the last kind of antibody is pretty much the most important antibody out of all of them. It's your IgG, okay? Now the reason that this is the most important is you can see that this accounts for 75 to 85% of your antibodies. So this is the most numerous of your antibodies that exist. Not only that, it does the most jobs, okay? Remember earlier I said that the variable region is what binds to your antigen, but the constant region is what gives it like the response. If you are an IgG, you have like all of the responses, which we'll talk about in just a minute. Okay? So if we look at this next page right here, these are the actual actions. So if we think about like IgM, IgD, IgE, all the other antibodies, they maybe do one or two of these. IgG, which is the most numerous, is going to do literally all of these functions, right? So it's like going to a store. Does the store only sell ice cream? Or does the store sell ice cream, cookies, cakes, cupcakes, all the good things, right? You want the store that sells the most things. That's what IgG does. Okay? So let's look at what all of those things are. These are words, you know I don't like to read to you. Here's a picture. Okay? So one of the first things that it can do is agglutination. We should already know what this is. It's clustering. Okay? So do you guys see right here how you have an antibody, and you guys see how it's like binding to this red blood cell, it's binding to this red blood cell, it's binding to this one and this one. So if you take a bunch of red blood cells and like attach them together, that's what the clustering is. So when you guys did those blood typing tests, that's what you were seeing right here. Okay? Now, in addition to agglutination, it also does something called neutralization. Now, let's say that this right here is your bacteria. And remember we said that bacteria fights from the outside, right? So it's an exogenous pathogen. Since it fights from the outside, that means that there has to be something on the surface of this bacterial cell that is dangerous. Okay, so let's say that there are these little proteins on the surface, and these are poisons. So it's like, remember how you were a kid and the ground was lava and you couldn't touch the lava, right? This is the lava. You can't touch this. If you touch it, you die. Okay? So the way that neutralization works is that the antibody comes in and if you have a bunch of antibodies covering each of these toxins, remember we said that the whole thing was you can't touch the lava, you can't touch the toxin. Can you touch the toxin now? No, they're physically being covered by this antibody. If you can't touch it, it's like putting like a protective layer on top, like protective piece of plastic, and now like you're not gonna get infected because there's nothing to infect you. Okay, so we're not killing anything. It's not like we're killing the bacteria is still there. We're just making it less toxic. Okay, any questions on that? Now, antigens can also do something called precipitation. Now, precipitation is a process that we talked about in blood clotting. You guys remember fibrinogen? How fibrinogen was, when it was in its inactive form, it was soluble. And then when we activated it, it became insoluble. You guys remember that? We went from like having salt water to having sand water. Okay? So the fact that we're making it like going from salt water to sand, that process is called precipitation. Okay? You're taking things out of solution where you can physically see it. It's no longer dissolved. So the way that this works is if you have these tiny little, like, do you see how those red dots? They're very small. That means that they're soluble. It's little salt water. But if you take enough salt and you connect all of them, like we're seeing here, it's going to take it out of solution and make it insoluble. Okay? So those are the three, or some of the major things that happen. We already talked about how it activates complement. Okay? Now, it can also increase phagocytosis. This is a process known as opsonization. Okay? Opsonization is just increasing the susceptibility for phagocytosis. Okay? So think about eating. Okay? If I have a cake, right? I go to the store, I buy some cake mix, I put it in a pan, put it in the oven, pull it out. If I pull it out of the oven, there's nothing on it, so it's just a plain cake, I'm going to eat it, right? But if I then put like icing on the cake, and then I put sprinkles on the cake, and then I put like chocolate pieces on the cake, and then caramel, and then all the good things, right? You want to eat it more than the plain old regular cake, right? You'll eat it either way. Well, I will. But you want to eat it more if it has the extra good stuff on it. The extra good stuff is this antigen, or antibody. Okay? Makes it more attractive to eat. And then we said that it stimulates inflammation. So that's that whole like IgE thing over there. And the whole complement thing, it can eventually lead to cell lysis. Okay? So all of this right here, look at how many ways we can kill or neutralize a pathogen just by like one molecule. Okay? So when we said that this right here is what determines what we bind to, but this here is what determines what type of response we get. The constant region determines, are you gonna do opsonization? Are you gonna do complement? Are you going to do precipitation? Are you gonna do agglutination? This tells you what job or how this antibody is going to work. Okay? Any questions on this? This is just talking about how IgE plays a role with your eosinophils. What is the eosinophil's job? Everybody gets their phone. At least it wasn't like going off everywhere. That's good. Okay, so eosinophil's job is to attack parasitic worms, right? But think about the size comparison. My board is getting out of control. I'm gonna have to erase something. Let's erase complement. Okay, so let's say that this right here is your parasitic worm, okay? Parasitic worms are huge. They can go like in like a big part of your intestines, right? Yes.